Hello and welcome to week 5. You made it through your first unit exam, and now we're going to dive deeper into one of the topics that was mentioned in that last week's cell, thinking about the membrane. But now we're going to talk about it much more deeply. So we were already slightly introduced to it, thinking about it as the bouncer that's deciding what comes into a cell and what comes out of the cell, because the plasma membrane is going to define what is in and out. And you were also already introduced to actually what makes up the plasma membrane, and those are phospholipids. So phospholipids you were already introduced to as having two parts that are very different. They have hydrophobic tails that are made up of those fatty acids, and they have a hydrophilic head that has charge based on the choline and that phosphate group, hence phospholipid, phosphate group with fatty acids. And the key for the structure of phospholipids is those two different sides, the one that likes water and the part that doesn't like water, means that when put into an aqueous solution, a whole bunch of phospholipids are going to self-form into this bilipid membrane, where those hydrophobic tails are going to come together and the hydrophilic heads are going to sort of protect them from the water, and they're going to interact with that charge, and these guys are all going to come together without charge. So you already knew about sort of the physical structure that makes up most of the plasma membrane, but we don't really know anything about how a membrane works. So we're going to now think about how does a membrane move? So we could do a little scientific method. We could come up with the observation, hey, I got two different types of cells. I've got a mouse cell that's one color, and I've got a human cell that's another color. So the proteins I can tell are different, and I've labeled them with different colors. I can ask the question, what happens when you fuse them into one cell? After the question, I can come up with a hypothesis, a testable statement. I could say the red mouse protein is going to stay on top and the purple human proteins will stay on the bottom. So you fuse them, I can still see the two different proteins and I cleanly see which is one cell and another. Or I could hypothesize that the red mouse and the purple human proteins are going to mix together. So I could then take my hypothesis and do the experiment. I fuse the cells. I look, how are those proteins mixing after an hour? I can then make my conclusion. What does this mean? This means that those plasma membrane proteins are able to move. So we think about how the membrane works, and we think about it as the fluid mosaic model. And if you wonder what does that mean, fluid, think about a fluid. Think about a liquid flowing. It's moving all around. Mosaic meaning made up of different things. Think of like a glass mosaic. So what are the different things that are in that membrane? Well, we got, of course, those phospholipids who are going to be able to move all around this way. And then we've got a bunch of proteins. So we've got these integral proteins that are going from the outside of the cell to the inside. This is the inside of the cell, which you can see by the cell structure. You can see the cytoskeleton, and you can see, of course, the extracellular side, the outside, with that extracellular matrix. So we've got proteins that are spanning from outside to in. We also have peripheral proteins that aren't going through that membrane, but are just staying inside the cell. And then what we can also see here are some of these proteins that have some extra sugars on them. Those are called glycoproteins because they've got some sugar or some carbohydrate, most likely glucose, but often other sugars. And that's on a protein. You can also have glycolipids, which are sugars added to a phospholipid. So that's the mosaic part, and the fluid is that it's all moving around. So you can watch this video, the lipids can move around, the proteins move around, but they don't flip so much from up to down. In fact, when they do, that's based on some signaling, and it's a signaling thing that's specific and meaningful. So little reminder about cholesterol acting as that buffer. So how the membrane moves is going to depend on those fatty acids and what's making up those fatty acids, if they're saturated or unsaturated. And they have the different properties of being solid if they're saturated and less solid if they're unsaturated, moving around. So you think about those phospholipids moving. So here again, we can see all the different levels of that bilayer. And then the question is to think about that bilayer as that bouncer. So it's deciding what comes in and what gets stayed on the side, what comes in and what does not. So what does come in? Those are going to be small gases, small nonpolar molecules. So what does that mean? You got to be able to get past all this area of no charge. And if you've got charge, you're going to get stuck around here with that water and that hydrophilic side. So we've got the things that do pass right through, but inevitably the majority of stuff cannot just pass through the membrane. So that's why you can think of the membrane kind of like the wall of a room. If we're going back to thinking about um, the cell as like a city, that's like the wall around the city that defines inside and outside the city. So it's not a great wall. There's definitely lots of things that can come in and out. And this is really important for how our cell can get gases to exchange for aerobic respiration. And if we think about what our lungs are doing, taking in oxygen, blowing out carbon dioxide, how that's all going down is those gases can just freely go through the membrane. But what can't are things with charge. So ions or large molecules, those we're going to see in a little bit are going to need these proteins. But small, non-charged molecules and gases, they can go right through. So if we were in class right now, I would get out some water and I would drop some food coloring in it, and we would all look at diffusion. Diffusion is something that you have seen before anytime you have ever added a color to water and mixed. You see that inevitably what happens if you were to add water or color to a water? It mixes. It goes from being highly concentrated to being evenly distributed. And we're going to think about these terms a bit more for equilibrium, but we already have the ideas. This is something that you already know just based on your life, that you can have a high concentration and you can have a low concentration. Concentration meaning how much there is of a thing in a particular space. And this concept of diffusion is the tendency for molecules to spread out evenly in available space. So what we're talking about here again is diffusion, where molecules are moving from high concentration to lower concentration. And again, where you can see concentration is how much of that thing is there in a defined space. And all molecules are going to move from high to low until they're evenly spaced out, which we already saw that word is for equilibrium. So looking at that a little bit closer, what does that mean in practice? So say you've got a barrier here, but there's holes, so things can go through, but you have one side where you've added some molecules of dye, the other without. What happens? Those molecules are going to bump into each other until sometimes they're going to flow through those cracks, through those holes, and they're going to keep bumping and bumping and moving and moving. Over time, that motion and hitting is going to keep hitting until they are evenly distributed. That even distribution is called equilibrium, where those molecules are going to go from high concentration of those molecules to low or high concentration to low concentration. And this can happen with one dye or one solute or many different solutes, where they're going to go for that particular solute for high concentration to low. And we see that this example of things moving down their concentration gradient is called passive transport, where no energy is used. And we're going to contrast this with active transport later. But before we get on to active transport, we're going to talk about passive, something that doesn't need energy, but that's special. It's special because it's about water. Just like we had our whole PowerPoint for water, we have a whole term for water moving across a selectively permeable membrane, or how water moves from inside and outside a cell, and that term is osmosis. So osmosis is diffusion of water. So what does that mean, osmosis? So we talked about how if you had two different concentrations of sugar and you had this open so that sugar could go back and forth, eventually those sugar molecules will evenly distribute in the water. But now we're saying something different. Now we're putting in a barrier that does not let sugar go through. It's selectively permeable, i.e. like the plasma membrane that decides what comes in and what comes out. Water is allowed to freely come in and out, but those larger molecules, they can't. So it's only water that can move from one side to the other. So we have to think about, where is there a higher concentration of water? Water will always move from high concentration to lower concentration of the water. But what that means is it's going to move from low solute concentration to high solute concentration, meaning there's more water on this side based on how much sugar, because there's the same volume, but there's sugar on this side and there's more sugar on this side. So more sugar, less water. So those water molecules are going to bump into each other until some of them go through these holes. There's going to be a net movement from high water concentration to low water concentration. That motion, that movement, will actually move the right side of this container up. It'll evenly distribute the concentration of the sugar by moving water from one side to the other. And if you watch this video, you can actually see water move up against its concentration gradient. So you can see if you have a membrane where it's only the water that can go through, you can actually see how it'll go over gravity. So again, diffusion is the solute moving until it reaches equilibrium. Osmosis is talking about the water, and osmosis is still going to have water going from high to low, but it's going to go from low solute to high solute concentration. So it's going to water the solvent from high to low. That's going to mean low solute concentration to high solute concentration. So we're going to talk a lot more about that, or I'm going to talk you through this scenario where I'm also going to be introducing to this idea of tonicity. So talking about how a water moves into and out of a cell, we have special names for it. We have special names to say, is the water going to come in or is the water going to go out? And to do this, we're going to be comparing. We're going to be comparing that bag or that cell to a solution. So just like I can't ask you, is Joe taller than, and expect you to have an answer, right? Because you need to know, is Joe taller than what? Is Joe taller than a 2-year-old? Is Joe taller than a refrigerator? Is Joe taller than a building? You don't know the answer to that question until you know what you're comparing it to. And that's the same thing with this term, tonicity. We're going to be talking about isotonic, hypertonic, and hypotonic. And the key thing for these is to be comparing. So if we have this cell that's at, say, a 2% sucrose solution, or in this case, a bag, that means it's going to be 98% water. If you were to put this cell into a 10% sucrose solution, or a cell into a 10% sucrose solution, that means you're going to be putting it into a solution with 90% water. What's going to happen to the water inside that selectively permeable membrane or selectively permeable bag? The water is going to go from high water concentration to lower water concentration. So the water is going to leave the bag. If I did the opposite and I put that bag of selectively permeable membrane into water, or 100% water, now which one's bigger? 100. So now the water is going to go, again, the water is going to move from high concentration into the bag where there's a lower concentration. The only other option is, what happens if you put a 2% sucrose solution into a 2% sucrose solution? Same amount of water, that water is going to be coming in and out the same rate. So no net movement. So no net movement means it's an isotonic solution as compared to the cell. A hypertonic solution compared to the cell is going to be, again, when the water is leaving, when there's a higher concentration of solutes outside. A hypotonic solution will be the reverse. So we think about the importance of these. It's really critical for all life to decide which water is coming into and out of a cell. And we're going to see how different concentrations are going to affect plant and animal cells differently. So if we have a hypotonic solution and you're an animal cell, where you've got water continually coming into and into that cell, with just the plasma membrane, that force of those water molecules is enough to burst the cell or to lyse a cell. But for a plant cell, that plant cell has that strong cell wall. Think of that cell wall like a chain link fence. But what does it do? It's going to provide the structure that when that central vacuole is full from all that water, it's going to be super stiff or turgid. And that's actually what a plant wants to be. That's where it's all upright. But when that same cell is placed in an isotonic solution where there's the same concentration of water coming into and out of it, there's no longer going to be pushing against that cell wall. That's when a plant is flaccid, or when it looks like it needs to be watered and the leaves are all droopy. But for our animal cells, without that, chain link fence or without that wall around us, that's where we want our cells, in an isotonic solution. So isotonic, same concentration of water inside and outside. A hypertonic solution, hypertonic is there's more solutes outside. And what does that mean? It means all the water is leaving that central vacuole, and that plasma membrane is going to shrink all in itself, and it's going to be plasmalized if it's a plant cell, or shriveled if it's an animal cell. Either way, it's going to be a very sad looking cell. But when you actually look at this under a microscope, I'll show you a slide of that next. Remember for all of these that we're talking about the solutions. So a hypotonic solution means it's a hypertonic cell. Isotonic are the same. Hypertonic solution means there's a higher concentration of solutes outside than inside. That's going to be a hypotonic cell, where the cell is going to be hypotonic and there's going to be a higher concentration of water inside the cell leaving. So again, here we can see a nice, happy, turgid elodea leaf where that central vacuole is huge and is pushing up against that wall, which you can just see these chloroplasts moving around because most of that space is just the water, and that's a happy cell. Whereas, I'll actually play this, you can see that central vacuole shrinking, and you can see the plasma membrane coming in on itself here. And why? Because all the water is leaving. So the water is coming out and those plasma membranes are coming in. The cell wall stays the same shape. Again, it's that chain link fence. It doesn't move. So we're making these comparisons thinking about a hypotonic solution is going to be in. And I always think about this because a hypotonic solution, the cell is going to be a big O. In a hypertonic solution, then you get those shriveled cells, which I think of like a skinny hyper person that's always like bumbling around and talking like this. They're all really hyper and they're moving all around, and so they're more. So hyper is more solutes outside.